Ever wondered how our genes function? Today, we delve into the fascinating world of gene expression. This biological marvel allows the information in our genes to become functional, turning abstract codes into tangible reality. It's like the conductor of an orchestra, guiding the symphony of life that plays out within our cells. This intricate process comprises two main stages: transcription and translation. Think of it like a two-step dance, each step vital to the rhythm of life. Transcription is the first groove, where DNA's genetic information is rewritten or transcribed into messenger RNA, thanks to the tireless work of an enzyme known as RNA polymerase. This sets the stage for the second step, translation, where this RNA serves as a template for protein synthesis. Each protein then plays a unique role in the body, from building tissues to fighting diseases. So the first step of this process is transcription. Let's dive deeper into this process. Transcription is the initial step in gene expression, a critical process where the genetic code in DNA is transcribed into mRNA. This fascinating process begins with an enzyme known as RNA polymerase. This enzyme binds to a specific region of our DNA molecule called the promoter. The promoter is like a start button, signaling the commencement of transcription. Picture it like this. Our DNA is a tightly coiled double helix, a bit like a twisted ladder. When RNA polymerase binds to the promoter, it gently unwinds the DNA near this region. This unwinding exposes one of the DNA strands, allowing it to serve as a blueprint for the synthesis of mRNA. As RNA polymerase traverses along the DNA template, it starts adding RNA nucleotides, which are complementary to the DNA strand. This growing chain of RNA, synthesized from the 5' to the 3' direction, mirrors the DNA template strand. However, this process doesn't continue indefinitely. It halts when a termination signal is encountered in the DNA sequence. This signal prompts RNA polymerase to separate from the DNA template, and the newly formed mRNA molecule is set free. Now, in eukaryotes, things get a bit more complicated. The initial RNA transcript, called pre-mRNA, has to undergo some modifications before it becomes mature mRNA. These modifications include capping, splicing, and the addition of a poly-A tail. In eukaryotes, the initial RNA transcript undergoes modifications before becoming mature mRNA. Let's understand these modifications. In eukaryotes, the initial RNA transcript, also known as pre-mRNA, undergoes several modifications to become mature mRNA. This transformation is a fascinating process that involves a few crucial steps. The first step is capping. This is where a modified guanine nucleotide is added to the 5' end of the pre-mRNA. The cap serves as a recognition signal for the ribosome during translation and also protects the mRNA from degradation. Next, we have splicing. Introns, or non-coding sequences, are removed from the pre-mRNA, and the remaining exons, or coding sequences, are joined together. This process allows for alternative splicing, where different combinations of exons can be joined together to produce different proteins from the same gene. Finally, a poly-A tail is added to the 3' end of the mRNA. This tail enhances the stability of the mRNA and aids in its export from the nucleus to the cytoplasm. Now that we have a good understanding of transcription, let's move on to the next stage of gene expression, translation. Let's recap what we have learned today about gene expression. This intricate biological process breathes life into the genetic code, transforming it from mere DNA sequences into functional outcomes. This journey from gene to function encompasses two main stages: transcription and translation. Transcription, the first stage, is a masterful process where the DNA's genetic information is transcribed into mRNA. This is orchestrated by the enzyme RNA polymerase, which binds to a specific promoter region in the DNA, signaling the starting point. As it moves along the DNA template, RNA polymerase synthesizes the mRNA chain by adding complementary RNA nucleotides. This continues until a termination signal is reached, releasing the newly formed mRNA molecule. In the world of eukaryotes, things get a little more complicated. After transcription, the initial RNA transcript undergoes several modifications before it matures into mRNA. These changes, including capping, splicing, and the addition of a poly-A tail, prepare the mRNA for its journey from the nucleus into the cytoplasm. Once there, it serves as the template for protein synthesis during the next stage of gene expression, translation. Through understanding these processes, we begin to appreciate the intricate dance of life happening within every cell. This knowledge opens doors to many exciting fields of study and research. So remember, understanding gene expression is not just about the science, it's about appreciating the complexities of life itself. Stay curious and keep exploring.